Dr. Pradeep Kumar completed his BSc Agri and MSc Horticulture from Kerala Agricultural University, Vellanikara. He obtained his PhD degree in horticulture from IIR New Delhi in 1994 and undergone overseas training at University of Wisconsin, USA. He is now working as Provost Head, Department of Vegetable Science, Kerala Agricultural University. He is also holding the additional charge of Vice Chancellor, Kerala University of Fisheries and Ocean Studies, Cochin. Dr. Pradeep Kumar has experience of more than 27 years in horticulture research, teaching, and extension as state and national level expert. He has developed 6 hybrids of different crops like watermelon seedless type, salad cucumber, parthenocarpic type, and ridge gourd. He was the editor of the Journal of Tropical Agriculture for 6 years and reviewer of Springer and elsewhere publications. He is the recipient of Krishi Vignan Award for the Best Agriculture Scientist instituted by Government of Kerala and Harbhajan Singh Award by Indian Society of Vegetable Science. He has published more than 82 research papers, 12 books, 16 book chapters, and 45 popular articles. He has received Indian Society of Vegetable Science Fellowship from Indian Society of Vegetable Science in the year 2016 and IAHS Fellowship from Indian Academy of Horticultural Science in the year 2019 and Indian Society of Spices Fellowship from Indian Society for Spices in the year 2021. With these few introductory remarks, I welcome Dr. Pradeep Kumar sir to start your guest lecture, sir. Thank you, sir. Thank you.
Very good morning to all. This is a nice custom followed in TNA so that you can use the expertise of the external examiner. And I will suggest this definitely in KU and other universities. So we'll go, this topic I'll be covering only my work. Okay, it's not the other works. What I have done for the last 28 years that I'll be explaining. Next, please. So I tried polyploidy, male sterility, gynaece, and parthenocarpy in developing new hybrids in cucurbits. So we know that the importance of cucurbits in food security and from the nutritional point of view, and in India, in all the states, perhaps the various cucurbits crops tops the list in production and consumption. Next. So coming to the importance of polyploidy breeding. We have, just to brush up your memory, we have autopolyploidy, allopolyploidy, and segmental allopolyploidy, isn't it? We know these terms. And there is naturally, there are 2-step or 1-step in development of polyploids. But we can induce polyploidy through artificial means. Next. And what is the consequence of polyploidy? The cell size will increase, leaf size will increase, giga size. And what are the application in cucurbits? You can develop triploid seedless hybrids. You can develop new species. And the fertility barrier can be overcome. And we can have tetraploids with a specific fruit quality. So seedless watermelon right from the days of Kihara. You know who is Kihara, Japanese scientist, way back in 1950. He proposed this technique and revolutionized the watermelon segment in world, I will say. If you consider the last 100-year development in vegetable breeding, this triploid seedless watermelon breeding is a major step. Other than that, we can consider what are the things? Male sterility in onion, male sterility in muskmelon. Okay. Before the genomic era, these are the classic milestones in the vegetable breeding. So we have, you might have studied Pusa Badana for all your JRF studies as studies. First, seedless watermelon. This was developed by introducing a tetraploid line from USA, from USDA, and that was released, but for the academic interest only. Nobody actually tasted that. Pusa Badana, we know the name, but after releasing it, it was not popularized by due to various reasons that we will see later. So in KU, way back in 2008, we started this program, and in 2012, we could achieve the first milestone. That is, we developed tetraploids, stable tetraploids by applying colchicine. Colchicine can be applied by different means. You can apply through seed treatment. You can apply it as a seedling treatment. Okay. For seed treatment, you should be careful because if the concentration is high, it will become sterile. And seedling treatment means you are applying on the leaf. You can go for a little bit higher dosage also. So this sugar baby was used. Why sugar baby? You know, sugar baby is a very old variety introduced in India in 1960. But the advantage is that now it got acclimatized under our condition. Means in Kerala it is high humid and high hot. So those hot humid condition now sugar baby is performing well. So we targeted sugar baby for developing this tetraploid stock. Okay, here you can see 0.5% colchicine seedling treatment. And this is just some data giving you, this is the C2 generation. When colchicine is applied, the first generation, second generation, instead of F1, F2, F3, a mutation breeding experiment, we call it as M1, M2, M3. When colchicine is tried, we call it as C0, C1, C2, C3, like that. So this is the C1, C2 generation. There is a, with respect to control, you can see that there is a difference in the size of the pollen grain. Next. And this is the fruits in the C2 generation. But you cannot jump a conclusion in C2 generation because in the evolution, there is always a tendency to revert back to normalcy. So the plant will revert back to diploid at any time. So from C2 generation again through selfing, you go to C3. Next. So C3 generation, again, you can see the comparison between C2 and C3. The seed size is now increasing. Next. Pollen grain size and the pollen germination also. The 4x plants, the pollen germination when compared to 2x plants, it is slow. These are all some indication. So the ultimate indication is you should count the chromosome number. Next. So this is the chromosome count. It's very difficult in watermelon. The meiotic chromosome number, root tip also we can study. Root tip is comparatively easy. So you know the chromosome number in watermelon, 2n? Anybody? 2n is 22. Okay. Oh, it is written there. Okay. Next. So this is the characteristics of C3 generation. Next. And we can, this is the most practical way. You can count in the chloroplast, this count can be taken, and this one between 4x and 2x you can count. Okay. Next. And after completing 6th generation, we tried the cross. And this 4x stock was crossed with the CL4, and we could produce chonima. That's a red flesh one. Okay. And the same genotype, that the same stock was, when it is crossed with the CL5, a seeded yellow type genotype, it produces yellow flesh mutant. So this gives you an indication of the dominance of flesh color. If you see the genetics book, you will say lot of gene symbols associated with the flesh color. And this is a particular flush color. We can see general yellow color is there, canary yellow color is there. Like that, lot of term is associated with the flush color in, thank you, flush color in watermelon. So here, this particular flush color, this canary yellow flush color is dominant on red. That's why the F1 produce yellow flush color. So what's the advantage of this female parent tetraploid stock? Because this is amenable for producing both red flush and yellow flush. Yeah, next. So to confirm that, we tried flow cytometric analysis also. This is the most precision way for analyzing the chromosome number count, ploidy level. So the ploidy level, we can see the clear peak between 4x and 2x here. And this paper was published way back in 2016 in Turkish Journal of Agriculture and Forestry. It is available, freely available in ResearchGate. Those who are interested can download it and go through the paper. Okay. So this is the seed production plot. And we are growing it under open, as well as it is also suitable for polio situation. I can show you a small video after completion of this presentation. So right now in India, from the public sector, only 3 hybrids are there. One is Shonima, red flush, other one is Swarna, yellow flush, and the other one is Arka Madhura, that is from IHF Bank. So these seeds are available. You can, those who are interested can mail to vegseedku@gmail.com, and they will guide you how to make the payment. Seeds are available, and we are sending seeds throughout India. Okay. There are certain limitations. Why still this triploid watermelon cultivation is not picking up in India? One is that the development of breeding stock. This tetraploid stock development is very tedious, and that maintenance, it should be under a strict control of a breeder. At any time, the 4x can revert back to 2x. And the seed production, you have to do the crossing under controlled condition. Okay. And if you go through the Amazon, right now you can see Google, see the seedless watermelon seeds, Amazon. In India, do you have any idea which company is marketing? Syngenta. Nannams is marketing one sector. Okay. And you just see the 10 seed cost you 100 rupees. So one seed, 10 rupees. So the huge seed cost. And there are a lot of patents. All are patented in US, patented information. So there are a lot of trade secrets involved in the seed production. Nobody will tell you. Now we gave this technology to 2 seed companies from Bangalore. They approached and we gave the parental material on non-exclusive license. Next. So this is the YouTube where you, the link, you can see the video. I can show you the video after the completion. This video is there in the, if you go through the Google, you will find it. So this is the exploitation of the polyploidy, triploidy successfully we did in watermelon. So next I will tell you about the exploitation of another system, very interesting system, that is male sterility. CGMS system is not common in cucurbit. In cucurbit, male sterility is reported in muskmelon, but that is GMS, genic male sterility, where the male sterility is in the nucleus. You know the problems associated with the genic male sterility in seed production. There will be segregation. You have to cull out the fertile plants before flowering. But if it is a CGMS system, as in the case of onion, we have CGMS system. So that is more easy for producing hybrid seeds. So that was the target. We could locate one male sterile genotype in ridge gourd. Next. You can see the difference in the inflorescence. See, the left one rudimentary anthers and the other one is plump. It's a fertile anther. Next. So when you find a male sterile one, the challenge is how to fix it immediately without changing the genotype. The moment you cross that particular plant with any other plant, what will happen? The genotype will change. So we have to go for tissue culture technique. So first of all, when we locate this male sterile plant, we maintained it through tissue culture, nodal cuttings. And then we crossed with a series of monoecious cultivar to find out whether fertility can be regained or the sterility can be maintained. Both are important. You know, the timing of anthesis in ridge gourd, that is during evening time. So during the evening time, this is the, you just see the comparison between the fertile, male fertile one on the right and the male sterile one on the left. So these are all the genotypes we use for crossing. Next. Okay, next. Next. Next, because there's a, you can go through the papers I published those things. Because the dominant fertility restorer gene is there. So there is a complementary gene action working out for the fertility restoration. Next. And we have find out a marker also. Next. So this is the two monoecious lines which regain the fertility. Means it is a fertility restorer. When you cross with the sterile line, these two lines produced fertile F1s. So we can use it as a fertility restorer line or as a heterotic combination. That is one finding. Next. Then another finding is that LA101, by crossing with the LA101, we could produce sterile one. Because when you cross it with the LA101, the F1 was always sterile. So that means that is the maintainer line. We can use it as a maintainer line. Next. So LA102, we can use it as a fertility restorer and LA101 as a maintainer line. That's how when you recommend a hybrid, you should have an A line, B line, and an R line. So we could develop both this. Next. So having very high yield potential. Next. Attractive dark green color. Next. This is the fertility restorer line. Next. This is the maintainer line. And these are the, this is the say 7.32 kg per plant yield. This is a very high yield and compared to control. Next. Fruit length, fruit number, and fruit weight. So this is a vigorous hybrid. Potential yield 36 tons per hectare, average 29. Next. This is in comparison with the Deepthi, our another released hybrid. So these are some of the publications from it. First one is regarding the tissue culture maintenance in current science. Next. This is in Scientia Horticulture regarding the dominant fertility restoration. Everything is available in the ResearchGate. You can download it. Next. And this is the exactly describing how the CGMS system can be used. That is published in U5TK. Next. And then how far this male sterile line can be successfully used for seed production. The ultimate target is seed production. So our male sterile line should be preferred by the honeybees, natural pollinators. So that work we published in Scientia Horticulture again in 22. This is also available in the ResearchGate. You can go through that. Next. So another important quality we are looking in cucurbits parthenocarpy. Parthenocarpy is important in almost all the crops from the consumer point of view. And what is parthenocarpy? We know it's the virgin fruits, means the development of fruits without actual pollination and fertilizer. Next. So there should not be any seed or traces only, or very reduced one. And we can have artificially induced parthenocarpy or vegetative parthenocarpy, as in the case of ivy gourd and cucumber. And we can have stimulative parthenocarpy and stenospermocarpy. I'm not doing because these are all things you have studied in your UG classes. So, what is the advantage? In polyhouse situation, we require parthenocarpy. So, there comes the importance of developing parthenocarpic hybrids. And once you taste the parthenocarpic hybrid, you will not prefer than a seeded one. So, very high quality, crisp, okay, and with a very good post-harvest life also. Next. So it's a boon for protected cultivation of vegetables, no need of pollinators. And in cucumber, this trait is exploited commercially. Just I told you the story of seed price of seedless watermelon. Like that, for parthenocarpic cucumber hybrids, farmers are exploited by the seed company people by charging 7 to 10 rupees per seed. So very costly seed. So what's the multi-pistillate gynoecious parthenocarpic hybrids are required now. Next. So these are all the sex forms in cucumber, and these are all the genes. Previous one. These are all the genes that can be exploited for the development of a parthenocarpic cucumber. And depending on the temperature and photoperiod, the original gene stock of the parthenocarpic cucumber is based on the temperate country. So those genes will not be stable under our situation. So what is our target? To develop a parthenocarpic breeding stock which is stable under high temperature and humidity. So next, we develop certain lines. These lines, both male as well as the female lines, are parthenocarpic, and this is maintained by applying silver thiosulfate. So silver thiosulfate induces male flower, and by that we can maintain the genetic stock without any change. So you should understand the science behind it. Both male as well as the female parent, both are gynoecious only. Means it will produce only female flower. You should not use a monoecious cultivar as a parent here. Once you cross it with a monoecious cultivar, the genotype changes, and it will be very difficult to recover the parthenocarpic lines from the advanced generation. So this is the KPCH1. This was developed in 2015. It's a very stable hybrid. Next. Now it is notified on all India basis, and this KPCH1 performs better under shade condition in open field also. Whether in coconut garden, arecanut garden, all people are cultivating it. These are the PPC 1, 2, and 3 are from Pannagar. We have KPCH1, and from IRA also we have some hybrids. And these are all the hybrids from the private sector. You can see there is not much difference in the color or the shape, but lengthwise there will be some difference. And like this, we have developed the gynoecious hybrids in Hira and Subra. Why gynoecious hybrids? Because it's very easy to produce hybrid seeds. Only gynoecious lines and monoecious lines alternately you can plant and allow open pollination. So artificial bagging and pollination is not required. These are Hira and Shubra. This is based on an introduced line, Gynoecious cucumber inbred. This is from Wisconsin only, popularly known as GY14. When it is introduced through NBPGR, they give this accession number, EC number. This is the Hira and the other one is Shubra. So Shubra is white and Hira is light green. But you cannot compare the quality with the KPCH1. KPCH1 is having superior quality. This one is having some spines and it will have seeds also inside. But this is suitable for open condition under rain shelter cultivation. Next. Okay. Regarding the yield, it is very high. You can see the yield of this 15.5 kg. Up to that 15.5 kg per plant, you can realize the yield. Next. Just a comparison. Next. So this is suitable for both under open and rain shelter cultivation. Next. See I told you there is some spines. Okay. Next. And now new 2 hybrids are going to be released in bitter gourd, again using gynoeci. So these are the 2 new gynoecious inbred lines developed in bitter gourd. We have inbred gynoecious inbred reported from IAVA Varanasi and IRA New Delhi. But those lines are with the small fruits, a dark green in color. But here the advantage is that here fruit size is comparatively large. And we have also developed the white fruited one, gynoecious one. So this is a huge potential in hybrid seed development in bitter gourd. Next. This is a gynoecious line in next. Yeah, this is a white fruited one. And this is not at release. Now it is under farm trial, showing very good potential. No, for protected cultivation, you require parthenocarp. Open. You can have it under open and rain shelter. But even then, nowadays farmers are growing even monoecious hybrids under protected system. They pollinate it, or by they install honeybee colonies. Yeah, since this hybrid is a subgynoecious character, the ready means they can grow it, realize high yield. So this is the fruit. You can see the fruit length of one hybrid. It's not at released. Next, and this is about the other hybrid. Okay, it's a highly heterotic and with the small spines, which is preferred in the central Kerala part. Next. So this is in general, just to give you an overall view of the novel hybrids developed from the Department of Vegetable Science. And I can say that for the last 20 years, I spent on the development of these hybrids. It's a continuous work. Sometimes I shared my story in my Facebook column. This ridge gourd. In fact, a ridge gourd master line I identified in 2002 from the field. But we developed the hybrid after developing all the A, B, C lines only in 2019. You see, 17 years it took. Namdhari is the leader of ridge gourd hybrids in India. And Northeast Kerala and Northeast India is the biggest growing region for this ridge gourds. And Namdhari now purchased this 3-line technology from KA. Because this takes that much time. You cannot just like within 3 years or 4 years, you cannot develop a novel hybrid. Just to show you, the kind of energy and the continuity you require for developing each hybrids. Okay, so I'll tell you that male sterility and gynaece and parthenocarpy are here to stay. We need to exploit these characters in other genotypes. Okay, so thank you. The green color of the gynaeceous line is dominant over the white color of Priyanka. That's the reason. So in general, if you see the genetics of bitter gourd, green color is dominant over white. But we cannot say exactly because you have seen the flesh color in watermelon. Each genotype, in breeding program, every time, the genotype is important. The quality of the F1 depends on the quality of the parent. You cannot say that yes, Preeti is white and Priyanka is white, so all the white will be recessive. We cannot take that line. As far as our study is concerned, this is the result.
Because my study in PhD also better good hybrid development only, sir.
Oh, good.
But I got two light colored rays also.
Hmm. But here, green colored one is preferred, no?
Yes, sir. Here, commercial is dark green and
Yeah, dark green color is preferred.
nowadays that poly is ruling, sir. Here, that gray tinge in the tip.
Ah, okay. If you are interested, I can show the video, or it's a 3-minute video. Otherwise, I will skip.
Yes, sir.
That is there in the YouTube. Last 5-6 years back, it is uploaded. Seedless watermelon is still a novelty in India. Unfortunately, in market we will not find many seedless watermelon hybrids. From Kerala Agriculture University, we have released 2 hybrids. One red flushed Shonima and another one yellow flushed Swarna. The seeds of these hybrids are now we are selling to the farmers at the rate of 30,000 rupees per kg, whereas the international price of this seedless watermelon hybrids is in the minimum of 2 to 2.5 lakh rupees per kg. Consider the seed cost alone. Minimum of 800 to 1 kg seed is required for cultivating 1 hectare. So you can just presume the importance of the seed price in the seedless watermelon cultivation. These hybrids can be planted in Kerala during the month of October, November, so that the crop can be harvested in February without much pest and disease attack. If you delay the sowing, then there will be more of virus attack and pest attack in watermelon. Watermelon can be sown either in the open condition or in the net house condition, both by traditional means as well as open precision farming. Here we follow the open precision farming, where the beds are taken width of 1.5 meter, and on the bed, seeds are planted at 1 meter interval. Here the most important thing is the pollinizer requirement for the seedless watermelon. When you cultivate the seedless watermelon, you need to plant 6 rows of seedless watermelon hybrid alternated with 1 row of sugar baby, which will act as the pollinizer. So after 35 to 40 days of sowing, the plant will produce both male and female flowers. Unfortunately, the male flowers of this Shonima or Swarna will not contain any pollen grain. So you need to pollinate for the female flowers so that the ovary will expand into a fruit. Under natural condition, honeybees will be doing this pollination, but when you are growing it under net house condition, you need to pollinate in the early morning through manually assisted pollination, collecting the freshly opened male flowers from the sugar baby and depositing the pollen grain on the freshly opened female flowers in the morning hours, preferably between 6:30 and 8. After pollination, fruits will be ready for harvest within 45 to 50 days. And on an average, you can get 3 to 4 fruits per plant, and we recommend only 3 fruits per plant, and the average fruit weight is in the range of 2.5 to 3 kg. From 1 hectare, we can realize minimum of 15 tons yield, and we can grow it under open condition as well as under polyhouse condition. So this is a good option for the enterprising farmers. And we all know that watermelon is a choicest fruit under in summer months with a very good market appeal. And particularly the seedless watermelon, if you harvest and go to the market, it will sell like a hot cake. So this watermelon hybrids, particularly this Shonima and Swarna, is a boon for the enterprising farmers in India, and KI is ready to part the technology with any stakeholder who is going to come for popularizing the seedless watermelon cultivation in India. Red flesh is popular. Yes. Some people like only yellow. Because the citrulline content is more in yellow. Quality wise, yellow is more good. Taste, this yellow, there is a slight difference in the taste. A little bit ash god like, a little bit sweetness.
Sir, I have one question pertaining to the citrulline content. So, is there any correlation between color of the fruit and the key molecular compound?
Yeah, I told you the citrulline.
Because in case of yellow, you mentioned that there is an increase in citrulline content,
Yes.
whereas it might have reduced. In case of Shonima, that is red flesh,
It is not reduced significantly, but it's in the range, but significantly citrulline content is high in
Swarna.
Swarna.
To what extent the difference might be, sir?
15 to 20%. That is the range.
15
difference.
to 20%.
TSS wise also Shonima is better. Shonima is having more sweetness compared to Swarna. But it depends on the growing condition. If you grow it under open, both are having very good sweet. But under net house or poly house situation, TSS content is slightly reduced. So the accumulation of this photosynthesis now under shaded situation, that might be giving this low TSS. The problem here is when so many farmers from Telangana, they brought the seeds. They are finding the pollination is the major problem. So you require an artificial pollinizer there. If you have this honeybees, they will take care, but they are not well versed in the farming techniques. Now most of the computer professionals are jumping into farming, and straight away they want to grow seedless watermelon. So what I advise them, you first grow normal watermelon, study the physiology for one season, then you start seedless watermelon. That also seedless watermelon, you can try it under 5 cents first, see what are all the problems, then you can expand. Now VFPCK also got this technology, and now they are going to popularize on a large scale. Yeah, this is a triploid technology. No, throw seeds only. No. Every time you have to purchase the seed from the. Yeah. Yeah. As it's a triploid male, it will be male sterile and it will be female sterile. So that doesn't mean that it will not produce flower. It will produce flower, but it will be male sterile and female sterile due to the faulty meiosis. Got it? Faulty meiosis, 33 only. So it will not, the chromosome will not separate evenly. That's the problem. There you come. Yeah.
While crossing two parents, how will you identify whether it is parthenocarpic or not in F2 segregating populations, sir?
By seeing the leaf shape or size or the vine length or the vine character, you cannot differentiate. You need to find out the parthenocarpic trait by tagging it and cover it. Otherwise, you cannot identify.
There is no very
No, 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 no. Nowadays, markers are being developed, and but most of the markers are QTL types. So, as I told you, for a genotype, it will work. For other genotype, it's doubtful.
If there are no doubts, then it's time to propose a vote of thanks. Uh, it's my proud privilege to thank Dr. Pradeep Kumar sir, who has come all the way from KAU Trishur to deliver a lecture on the innovations and hybrid development of cucurbits. So here you have got some 27 years of experience, and everyone, the students are really delighted to have your lecture, sir. It is like a hot cake. You have shared 27 years of your experience, and it's really our honor. We are honored to have you here, and your hear the experiences on the cucurbits especially. Especially you have explained all those tetraploid varieties, male sterility, parthenocarpy, and gynocy. And we are much privileged to see your publications as well. It is highly motivating to our students that we should have such a publications. We should leave it for the future generation. Like so, thank you so much, sir, for being here with us today. And I thank all my staff members, especially the then PG coordinator. She was very, Dr. Tangamani, she was very particular that we should invite sir for the Viva OC at any cost. Like she has taken all the initiatives in the beginning, and then it was continued by Dr. Haripriya, the now PG coordinator of the department. And I thank all the staff members of my department for participating in the guest lecture, and my students, so who are always there for us to support us, to take the experience of user. Thank you so much, sir.